Hey guys, Nurse Oz here. We are going to demonstrate how we are going to take some of our bacteria that we grew in lab last week, and we're going to transfer a small amount of this to a microscope slide so we can view it under the microscope and look at it, each of the cells individually, okay? So the first thing that we're gonna do before we get started is we're gonna look at the cell morphologies or the colony morphologies that are on this petri plate. And we're gonna use those to describe what we see in each of our quadrants that we made, okay? So you're gonna do that in class before we do this step. So once you've done this step, then we can get ready to transfer this stuff to the microscope slide. So we're gonna make sure that this is nice and covered when we're not using it and just place it off to the side. We're gonna take a clean microscope slide and we're gonna label it with what quadrant we're taking it from. So I'm gonna use the one with the most bacteria, which was fingers from a student that accidentally touched the agar with her fingers. So we're gonna use that. We're gonna label it fingers, okay? And then we're going to take a very small amount of water from a pipette and we're gonna add it to the slide. Okay. Now we're gonna take a Q-tip, okay, and we're going to take a small amount of the microbe that we want to look at under the microscope. So we're gonna just take one of these nice little cells here. We're just gonna scrape off a little bit here, just enough to that you can see it on the tip of your toothpick, okay? And we're gonna add it to the water and we're gonna swirl it around and make sure that it gets nice and diluted on the slide because we don't want to see big clumps of our bacteria. Otherwise, we're just gonna see purple stuff and you're not gonna know what that is. So we're gonna spread it out on the slide evenly. And you want to spread it a lot because if the water is clumped up at all, it won't dry. It'll take a long time for it to dry. Now, typically, if you were working in a microbiology lab, you would do this with a sterile inoculation loop. But we don't have that, so we're improvising with toothpicks. Okay, so now you can see that my slide is nice and opaque underneath there. I don't know if you can see it very well. Under where I placed that stuff. So you can see that kind of filminess there. So we're gonna let this dry, okay? And once it's dry, then we will dye it with methylene blue so we can see it under the microscope. So when this is dry, we'll come back and we'll stain it. Thanks, guys. Hi guys, Nurse Oz again here. Now our slide is nice and dry. You can hold it up to the light and make sure that there's no wet spots on this slide, which there's not anymore. So what we're gonna do is we are going to take our slide and we're gonna dye it with some methylene blue solution dye, okay? And this will turn the bacteria a very bright purple color. So when you look at it under the microscope, you will be able to see the different shapes and the cellular morphologies of each particular bacteria under the microscope because this will turn it nice and purple. So what we're gonna do is we're just gonna add a few drops to our slide. And we're just gonna kind of dye the center of it because we only need to look at just a small, tiny little area in the microscope. So you can take your slide and kind of move some of the dye around if you can. It's kind of finicky. So it doesn't have to be perfect. As you can see, it leaked all over my glove, but that's okay. So we're gonna wait just about a minute for this. And then we're gonna rinse it off with some sterile water, okay? I guess it's not sterile, it's distilled water. So we'll wait just a second here. A few more minutes, seconds. All right, so that's been about a minute. So now we're gonna take our slide and we're gonna hold it at an angle like this, okay? And then we're just gonna gently spray the water on there. And you guys can spray on the Chex pad, it's not gonna make any difference, okay? So you're just gonna gently dab the edge of the slide on your Chex pad, okay, so you can get the excess water off. You can do it on the side, okay? Now, it doesn't look like there's a lot there now, but there is a purple tinge to this. So that means that the bacteria are dyed good enough that we can view them under the microscope, okay? So we gotta wait for this to completely dry. And we have two ways of doing this. You can kind of just put it on your Chex pad. Because normally in a microbiology lab, you would heat fix this after you dyed it and after you place the bacteria on there, which we don't have Bunsen burners or any of that fancy stuff. So we're just gonna let it air dry, or you can take a little bit of paper towel and just gently pat it dry, okay? You don't want to rub back and forth, okay? And just move it till it's dry. Now, typically in a microbiology lab, you would use bibulous paper, but again, I don't have bibulous paper, so we're improvising, and this is what we're going to use. And then this is what your final product will look like when you're done. As you can see, you can still see some of that purple tinge to the glass, which means our bacteria got dyed properly. And that is how you prepare a slide in my lab class this week. Thanks guys for joining me. We'll see you in lab. Thanks. All right guys, so here we are at our lovely microscope, and now we're gonna look at the bacteria that I just dyed in the slide and on the last video, and we're gonna take a look here at our slide in the microscope and see what we can see. So as you can see, and I'm sorry for the blurriness, we have, looks like bacillus rods, and they're in chains and they're clustered. So that's how you would describe the cell morphology of these. We do have some artifact in here just because we did not use bibulous paper and we didn't use the sterile inoculating loop. But that's okay, you get the general idea that we have rods that are growing in this particular slide that was on my students' fingers. So, cool beans, pretty cool. Let me know if you have any questions and we'll talk to you later. Thanks guys.